All right, guys, let's get started. So a few course notes. For the homework, I don't mind you guys sitting together and working together and talking about the homework questions, but please don't copy. If I see like word for word answers or like scripts turned in that are exactly identical, I'm going to be suspicious. So try to like walk that line where you're working together, but you're not copying. About 6 of you had your quiz grades disappear. I think it was homework 1 was the big problem. And I went back and forth emailing with OIT. And what happens is that if you start a quiz and submit it, and if at any point you start another one later and don't submit the later one, it deletes your first one. So don't try not to have like unsubmitted versions of the quiz. And it's hard, I don't know exactly what it looks like from your perspective. So if you see that you have multiple versions, try to make sure you submit the most recent one. Because if you submit one that's earlier than the most recent one, it causes the most recent one to submit and give you a 0, which is bad. And we rescued all the grades for like the 6 people whose quiz grade got lost. I think we got the correct grade restored. But just a warning on that. And if a homework grade disappears, let me know, because I won't necessarily notice unless you email me and say, hey, my 10 just became a 0, fix that. So definitely let me know if that happens. Everyone who has an iPhone, or lots of people who have an iPhone, if you're getting security problems logging into the ACF, and I'll show you when we log in today, you can do a push, and that seems to fix it for everybody who isn't totally locked out. Like Rob. I think everybody else, it's going to fix it for it. Yeah. Yeah.
Up to now, if you want to retake one question,
yeah, I don't think that that's a Canvas feature. I think you have to redo the whole thing. You can't redo just one question. And I had this problem with somebody else who like submitted the first 8 questions and then she couldn't go back and even look at her answers. So I like downloaded the answers and sent them back to her so she could like put them back into the next version of the quiz. But if you just don't submit it, you can go back to it. Just don't submit it until you're ready and do it, because it'll get saved. And then just don't submit, if that makes sense. And then you can keep going back to it until you're ready to submit. And I think that fixes the issue. But if you start a new one and then submit the old one, that's when we run into problems. Or if you submit one and then you can't go back and see your answers, that is also a problem. So just you can keep going back to it, but don't submit it until you've answered everything and you feel good about it. Yeah.
You can possibly post the answers to the homework, so we can
yes. Yes, absolutely. And I tried to go through and comment on everything, and I don't know how to do that on Canvas yet, but I will try to figure it out. Canvas is very special. I didn't love Blackboard, but I like it a lot better looking back now. Yeah, I can do that. Any other questions about the homework? I posted another homework that's not due until next week. It's due next Wednesday. Yeah, so I want to go over a couple of things that seem to be like widespread things that we could, kind of like 2 things that I think we could work on that'll make everybody's life easier. So somebody asked me, I have this little shortcut set up where I just type SSH ACF and it knows where the ACF is, and I'll have to type all that, and I will show you how to do that. I'm going to post it though, because it takes a little while to set up, but I will post instructions before the next class period on how to do that for those of you who are interested. Okay, so this is where if you're having problems with your phone and you normally look at your phone and you look at the number and then you type it in, don't do that anymore. Press 1, hit enter, and then your phone should give you a green button that will get you around those security hazards if you're not ready to update your operating system. Because right now, if I type in that little code, it won't let me. Okay, so if you have problems, further problems, email the ACF. help desk, and hopefully they'll help you get it resolved. Okay, so tab complete. This is something that maybe like a quarter of you were actually doing in the shell test, which is really sad because this is going to save you so much time. So let's go look at our course website. Oh, that's pretty big. This one. So we're going to end up typing this in a whole lot, and that's a lot of letters, and it's a lot of things to misspell. Like Lester's not even spelled like a normal English word. So what I do is CD/LU tab. And if you hit tab and nothing happens, that's not a problem. It's not going to ruin your command. Like I know the next one's Haven, so I'll type H tab, and it fills it in. And I'll type C tab, and it fills it in. And I'll type capital E tab, and it fills it in. So it's like, it's just gonna save you so much. Like just start hitting tab all the time. And then my username starts with an M. So I'm gonna hit M tab. And it's not gonna fill it in, probably because there's somebody else with the username that starts with M. So I'll type the next letter, MS, tab. Okay, there's probably someone else with the username MS. And then I'll type T, tab. Okay, it found it. So I have this tendency when I'm typing paths to be like a letter, tab, a letter, tab, a letter, until it fills it out for me. Because, and that's also just going to prevent you from doing so many misspellings. It's going to save you so much time. Because I don't even have like EPP622-2018 FA memorized, because I've already figured out that I just type capital E tab and it works. So it's just going to make your life easier. So practice that. Okay, the other thing that I think we're still having a lot of problems with as a class is this whole like, the root of the file system is a backslash. So if I want to say, I have this test.fasta. So if I say head test.fasta, it is in my current working directory, so I don't have to put anything in front of it. It's just going to give me the head of that file. But if I put head slash test.fasta, that's not a thing, because that's saying nixd home. Might not have it here. So slash is always that top folder. So my actual file is way down here. So if I put slash file, it thinks I'm saying that it's up here. And that's why it's like, I don't know what that is. Because it doesn't exist here, it exists here. So my two options are to say the relative file path, which I can also do like this. So by putting this period, that anchors me to this directory. Inside of this directory is test.fasta. Or I can do the whole giant path, which is head, nixd, home, mstain1, test.fasta. And that will also work. But if I do head, test.fasta, that is not a location, that file does not exist. So is that like if you put a backslash as the very first thing in a path, that implies that whatever you're doing has to be, whatever you're pointing to has to be in here. So like nix is in here, so slash nix will work. The slash test.fasta won't work because that's not in there. Does that make sense? Okay. If there's any questions, let me know. I think that was like one of the most common problems, so I just wanted to go ahead and talk about that. Were there any other like things that you guys want me to go over as far as basic shell commands? The tab?
No.
Oh, Q sub?
Yeah.
Okay. Yep, and we're gonna try it today, and I have no idea if it's gonna work. I've been emailing back and forth with the ACF, and this is what I think is supposed to work. we, as a class, I think like 4 people got it to work when we tried the class period before the shell test, or like the last time we were actually met and had a class. So the goal here is when you log into the ACF, you're on this login computer. And this login computer, like everybody from the whole university, we're all logging into this. So if we all tried to run our jobs on this, it would get way too full and it would probably crash. And that would be bad. And I don't want that to happen. But we all have to have like a common place to go in. But this is running the software that figures out how to launch jobs on, you know, the thousands of other computers that they have. So we go in and Q sub is basically, that stands for submit, and Q, so we don't use the word queue in this country as much, but in Britain the Q is a line. So if you get in a line for something, it's a Q. And it's actually spelled Q-U-E-U-E, but it sounds like the letter. So I'm submitting something to get in a line is what that command means. And by the rest of the command is saying, I want an interactive session. I want it to be billed to this class project. The specific line I want is the debug queue. So it's kind of like lines in the grocery store where there's like lane 1 and lane 8 and lane 10, and you pick which one looks fastest. We think the debug queue is consistently going to be fastest.
How do you check the list of lanes that are viable? Like, you may pick
yeah.
busy.
You have to read the ACF documentation to see all the queues that are available. But I only specify the debug queue for this class because it should be significantly faster than any of the other ones. They've set aside resources for us specifically on the debug queue. And so everyone who is part of that project and so does this exact command should, quote unquote, should get, so it'll pop you over. So now your SSH, it's like tunneling. This is called tunneling. Because you're not talking to this computer, but this computer is like forwarding all your requests over to this computer. And so like I might be here, and Rob might be here, and Camille might be here, and you know, like we're all gonna be on different computers. And that way our resources are being like, our requests are being distributed across all these different computers. So we're not all running into each other and fighting over the same login computer. So that's what that QSub is doing. Does that make sense? And so if you ever hear the term in computer science tunneling, that's what it means, is you're like logging into this computer and then logging into another computer, so you're tunneling through it and just creating a little, this isn't gonna take much resources from this computer because it's not doing anything except receiving your command and sending it on. So it's just like a pass-through.
And the rest of the command all time?
Yeah, so unfortunately the maximum wall time for a job they would give us for this class is 1 hour. On a different queue, like on a regular queue, I think you can have up to 24 hours, but you're supposed to estimate, like I think this job is gonna take 12 hours, I think this job is gonna take 20 hours. And you put in an estimate of how long you think you need. If you put in an hour and your job isn't done in an hour, it just kills it and drops you off the computer. The tunnel collapses and you're done. So it's important to overestimate. It's kind of important to estimate well, because the longer your job is, the harder it is to schedule, and the longer it'll take to get run. But if it runs in your... Why?
I don't see any reason.
Well, because if it has something that needs a whole day to run and needs 10 cores, it knows it's going to tie up this computer for a whole day. It may pair it with other whole day jobs. Whereas this computer, if they can keep cycling through lots of 1 hour jobs, it's like if you're trying to build your schedule and get lots of things done all in parallel, it's nice to know what things are going to take a lot of resources in a long time and what are going to be short and fast, so you can kind of fill them into the gaps. All computer schedulers require you to give a minimum time, well, all that I've ever used. So, or to give a time, and then it just kicks you off at the end of that time. And it is a trade-off, because if you launch a job that asks for a long time, it takes longer to get scheduled. But if you ask for a job that takes a short amount of time, it's faster to get scheduled, but if it dies before your job is complete, then you start all over. So it's a bit of an art. And then the other thing is nodes equals 1. So if you were launching something really crazy that needed like 10 or 20 or 50 computers, you can tell it you want lots of computers. We aren't going to need that in this class. That's the whole QSub command. We'll see if it works today. If not, we're just going to beat up on the login node again and see how long it takes them to kick us all off. But I emailed them and told them it didn't work at the last class, and they didn't respond. So I said we would try again on the 19th and to get ready. They didn't respond to that either, so we'll see. But good question on the QSub. Any other questions? No. All right. Okay, so I have some slides. Not that. Okay, I want to talk a little bit about applications of DNA sequencing. I also will talk a little bit about reference genome availability. When you guys decide on final projects, the difference between having a reference genome and not having a reference genome will completely change how you do all of your informatic analysis. So it's good to go check and see what kind of reference genome you might have available for your system. And then I'll talk a little bit about how to do quality assessment and trimming of DNA sequence reads, and then we'll actually do that for the lab. Whole genome shotgun sequencing is most of what this class will be covering, and it's an extraordinarily popular technique right now. Most of you are in this class because you want to use this technique. But in general, you start with genomic DNA, you shear it into fragments. There are a number of different ways to do shearing. It can introduce bias into how your sequences are generated, but there are physical, enzymatic, and chemical ways to do this. Most of the companies we work with are using Covaris, so kind of a pretty actual legitimate random fragmenting of your DNA. This can also be dependent on the quality of your DNA when you would extract it. So these are all things that you want to talk to your sequencing company about and at least understand when you get your data back, like what was done to my DNA to get it into fragments. But somehow it needs to be put into fragments of a reasonable size. Ideally, it is as uniform as possible. The informatics is easier if you know how big those fragments are. So depending on the kit, 200 to 600 bases is a normal size. A really tight selection might be like exactly 400 to 500. And I talked about a little bit a few class periods ago. You can have much larger fragments, like 3 to 20 kbp, but those would be called mate pairs. This is commonly what you're going to see for a paired N kit. So starting from there, it seems like, oh, okay, we're all starting from the same place. The analysis is going to be the same. But it turns out there's lots and lots of things you can do with this like one basic little laboratory protocol. We have turned it into like a ton of different amazing biological assays. There are 3 basic things that I think you usually want to do with your DNA sequence data. The first one is a resequencing analysis, where you have a reference genome, you kind of know what your species looks like on a genome level, and you want to find differences. Whether that's, and we'll go through, there's a ton of different types of differences you might want to find. The most common is probably SNPs or short indels. So this would be if you want to look at like population structure, or if you want to find a mutation in a gene of interest, or if you want to do phylogenetics, or whatever you're trying to do. You're starting with a known kind of what your genome looks like overall, and you're mapping reads from a different individual back to that reference genome. The second common thing that people want to do with this data is count it. How many times did I see a thing? The most common counting application would be RNA-seq. So you want to say, how many times did I see this gene being expressed? You could also do it for copy number variations. How many times is this copy in the genome? But this also is generally dependent on a reference genome or a reference of some type. So you're mapping the reads, and instead of looking for how the reads differ from your genome, you're just counting the number of reads for each location in your genome. The third one is an assembly, which is where you don't have a genome, and you're trying to, or transcriptome if it's RNA. And so you're starting from scratch, and you're trying to take all these little tiny pieces and put them together. So these are This is how every genome starts out. It has to start out as a pile of sequence data that gets put back together. So these are the three main things, and we're going to see all of them in this class. But these are really basic. It turns out that that does not at all cover the broad spectrum of amazing things you can do with sequence data. Because biologists are clever and can do amazing things and with molecular biology, we've really kind of turned this into a ton of different types of omics and a ton of different type of assays. So depending on how you treat the DNA and how you prepare the library, these are all things you can do. So you can extract RNA instead of DNA, convert the RNA to cDNA for sequencing. We'll cover that as like a whole section, separate section of the course. You can do targeted DNA sequencing where you're only sequencing regions of interest instead of the whole genome. ChIP-seq is great. You can find genome-wide binding sites. So for example, if you have a transcription factor and you are really curious where does this transcription factor bind in my genome? You can do these chromatin immunoprecipitation, you use antibodies, and you pull down just the parts of the genome that it's binding to and only sequence those. I'm going to talk a little bit today about GBS and RAD-seq, which are both ways for large-scale genotyping, where you don't want to sequence the entire genome, you just want to sequence part of the genome in order to get variants. We can now do methylation sequencing, particularly with bisulfite, where you treat DNA and figure out where methylation marks are on your DNA. There's Hi-C, where you can figure out the three-dimensional structure of DNA, but all of these are based on shotgun sequencing. So all of this is really manipulations that are going on at the DNA extraction and library preparation level. When that is dumped onto the sequencer does the same thing that it always does. It sequences those little fragments and spits them back out. It's important to understand all of this because your informatics analysis is completely dependent on which of these you did. All applications are dependent on appropriate coverage. And I thought I should actually talk about what coverage is. Usually in shotgun sequencing, you're thinking about depth of coverage, not breadth of coverage. I don't know if that makes sense. But we want to look at the average number of reads that cover a reference base. And that it's a super easy calculation. You take the number of map bases divided by the number of reference bases, and you know exactly how many reads are covering on average each of your reference bases. This is often displayed as a histogram because your coverage uniformity matters too. So let's think about coverage for just a second. And this is, I'm going to use SNPs as an example, but it turns out like the statistics in every type of informatics analysis based on sequencing is going to utilize coverage. The more coverage you have, the more confidence you have. So just looking at this by eye, we have these two sequence reads mapped back to our reference genome. Do you think that our individual that we sequence actually has a SNP at this fourth base pair? We've only got one read. We know there's 1% error between, around about 1% error for our Illumina sequences. Is this an error or is this real? We don't know. The same thing with this A. Do we have a heterozygote if we're working in a eukaryotic system? Is this A real? We've only got one read. Statistically, you're not very confident. Here, I've got two reads that cover the space. They're both C. I'm starting to get a little more confident. Over here, I've got 5 reads that cover this one base in the reference. I'm starting to get very confident that this is a real phenomenon. So whether you're doing methylation-seq or GBS or targeted sequencing, think about coverage. Before you order your sequence data, think about coverage. Think about, what do I need to do the statistics that are going to get me to my answer? Talk to a statistician before you do sequencing. I frequently have people come to me that have, not frequently, but occasionally people come to me, they've got no biological reps, they've got insufficient coverage, and they're just hoping, because they already blew their entire sequencing budget, that they can do something with it. And the answer is generally, you can't really do anything with that. Yeah.
So how do you know if out how your test is due to low coverage, or if let's say gene is lowly expressed?
Oh, for RNA-seq?
Yeah.
The more lowly expressed a gene is, the deeper you have to sequence to make it statistically significant. So generally, it just won't be significant. And whether or not it's real is, right, like statistics can't tell you if something's real or not if there isn't enough data. It can only tell you that it's not significant. And so with RNA-seq, that turns out to be a huge problem with very lowly expressed genes because you have to sequence so deeply to get enough data to do the statistics on them. So I think the ENCODE consortium suggested, I won't get the numbers exactly right, but it's something like 10 to 20 million reads per sample will get you enough quantity for about half of your genes of interest to do the statistics properly. If you want to get up to like 90% of the genes in the genome, so the ones that are more lowly expressed, you have to increase your sequencing depth by a factor of 10. So it's like you go from like 20 million reads to 200 million reads per sample in order to get, there's like this chart that they published. It's kind of old. It's from like 2011, 2012. But they had this chart they published where you could look at the percent of genes that you want to be able to accurately assay and like the amount of sequencing you need. And because lots of genes are very highly expressed and you can nail those with a little tiny bit of sequencing. But as the genes become less and less and less expressed, you have to do more and more and more and more and more sequencing per sample. So you just have to dig through the literature and you can find those papers that'll tell you that. But yeah, it's a hard problem. Yeah. That was all for human. So again, that's going to depend on your organism. If you're working with bacteria or yeast, you might not really have that many genes. Or if you're doing DNA, your genome is quite short. But if you're doing something like pine trees that have a 30 gigabase genome and you're doing DNA, then you're in a whole lot of trouble because that denominator for is very, very big. So you have to increase your sampling, increase your sequencing. So again, it's just highly organism dependent and application dependent, right? And you have to think really carefully because with RNA-seq, you're able to count the reads and it's pretty straightforward. But for something like variants, let's say you're going from, instead of using a diploid, you're using a hexaploid. Well, if you want to know 1 out of your 6 alleles is different, you have to sequence a lot more depth to get to a statistically significant, I could detect if 1 out of 6 was different. Or with bisulfite sequencing, where the methylation might not be penetrant in every single one of your cells. So you're looking at the ratio of methylated to unmethylated sites. It's going to be not consistent in your cells that you use for sequencing. So all of this turns out to be quite application dependent, which is a good question because that's the whole point of today's lecture. This is how you would traditionally see depth of coverage as a histogram, where the coverage is along the bottom and the proportion of contigs that met that coverage is along the side. The Illumina website always has this like perfect normal distribution of coverage, which is totally not ever going to happen in any of your sequence projects. But what do you think causes that long tail? So the very, very small number of contigs that have really, really, really high coverage. Any thoughts? Yes. Yeah, so repeats, if repeats have not been properly sorted out in your reference genome, if they're collapsed, you're gonna get, like maybe there's actually 1,000 of this repetitive element in your genome, but the reference genome only has 50 copies because they couldn't assemble it properly. You'll get tons and tons and tons of reads mapping because it's actually, you probably have the same amount of coverage, say like 15 over the repeats. There's just so many of them. And you're building up so many reads that they look like they're all covering the same region of the genome. So yeah, that long tail, which is never in any of Illumina's slides, is usually caused by repeats. So briefly, a little bit about whole genome resequencing. This is where we're assuming that the reference genome is already available. We're gonna look at this and starting today, we're gonna do a little DNA-seq. They're gonna do like 4 class periods with the same data set. And it's with E. coli, I think. Yeah, E. coli, because we have a reference genome. So for those of you that have a reference genome, congratulations. That's really great. And it's going to make your informatics a little bit easier. You're usually sequencing from the same species. If you go too far afield with different species, the reference genome isn't going to be close enough to your genome for the reads to map. So that's something to be aware of. I just had a postdoc kind of write a publication on like, what is the distance you can use for a reference genome? It turns out certainly you probably want to stay in the same genus, at least in plants. Same species is better. And the goal here, at least the one we're going to primarily talk about in this class, is discovering variations between the sample you sequence and the reference genome or among your samples. So the straightforward ones that are pretty easy to do are looking for single nucleotide polymorphisms and short insertions and deletions. It is harder, but possible to do larger structural rearrangements and copy number changes. And we won't go through this in this class, but if you're interested, I can point you to some software packages. But the bread and butter for most sequencing projects are these SNPs and short insertions and deletions that can be captured within a single DNA read. So you often hear in informatics like pipelines, what's your informatics pipeline, or what's your workflow? And so the Linux lessons were kind of a good example for this, because we saw how you take head and use that pipe, and you take the output of head and stick it into another program like sort. And the pipe is literally taking the output of one thing and shoving it as input into the next thing. And that's very much how most informatics works when you're doing analysis. So kind of the example that we'll be going through is getting raw data, looking at quality assessment of that raw data. Is it okay? Am I good to use this? Or should I call the sequencing company and ask for my money back? You know, that's a good thing to know. Trimming, so getting rid of adapters, getting rid of low quality bases. I suggest doing your quality assessment again to make sure you fixed the problems that you saw. Especially if you saw adapters, go ahead and redo your quality to make sure the adapters did indeed get removed. Take those clean reads, map them to a reference. At this point, a lot of people skip this step. I don't recommend it. Go ahead and visualize, like load all your data into a way where you can look at the reads, how they're mapping to your reference, what kind of coverage you have. Are the numbers that the computer is telling you lining up with what you're visualizing and actually seeing as far as the alignment? Most people skip that step, and it's generally a bad idea to skip that. Next, actual calling of variants, finding those SNPs or indels. Next, we'll see really briefly, there's a couple of nice software packages to assess the impact of the variants if it occurs in a gene. Does it change an amino acid? Does it alter a splice site? Does it create an early stop? So it's kind of nice to know which of your variants might be functional. And then we won't go through this again, but it's nice to always remember at the end of the day, you're gonna have to submit everything to the sequence read archive in order to publish. So again, pipeline, input from one thing is becoming, or output from one thing is becoming the input to the next step. And you're doing things in a reproducible order and keeping track of exactly how your data is organized and being analyzed. Some other apps that we're gonna see in this class. We just hired a new professor, Dr. Bode Olukolu. He's great. He is a very brave person because his thing is genotyping by sequencing in polyploid plants, and he can actually sort out allelic dosage and hexaploids. He works on sweet potatoes and maize, and that's really cool. And he's going to do 2 lectures about how to filter, how to call variants, and how to filter them appropriately. And his work has revolved very much around this application, which basically polymorphism discovery, very important. You can use it for all sorts of things: population structure, association mapping. He does a lot of association mapping in particular, pedigree mapping, QTL mapping, phylogenies. And this has become really popular as far as replacing SNP chips and microsatellites because it's cheaper. It's become cheaper. But it's still not quite cheap enough because whole genome resequencing is expensive. I was just meeting with interview candidates for the genomics director position over here. And one of them told me that to resequence an entire cow right now is still over $1,000, but to do genotyping by sequencing is less than 100. So they're getting per cow, from extracting the DNA to sending it off to sequencers to getting their variants back, they're less than $100 versus a very expensive over $1,000 to get the whole genome. So basically, this is a way to vastly reduce the cost also vastly reduce the amount of data you're producing. And it works like this. So I'll start with RAD-seq because I think it's the original. This is a random genome reduction technique. It was developed in 2008. You want to be able to identify and score thousands of genetic markers by subsampling only at specific sites. You use restriction enzymes to find specific sites in your genome and prime and sequence off of those restriction enzyme cut sites. So instead of trying to sequence everything, you're very specifically looking for these restriction enzyme sites. They are quote unquote randomly distributed across the target genome. It turns out they're not really randomly distributed at all, but they're random enough. And you can do this for many, many individuals and then use your Illumina technology to do the sequencing. This whole process was patented. As far as I'm still pretty confused about this. I think there are a number of places that license it and offer it. The big ones are Keygene and Fluorogenics. A few universities also offer it. I don't know if the universities have to license it or if nonprofits can use it for free. I've gotten kind of conflicting information on that. It's not something we offer as a service at this university. So, but I used to contract with Cornell to do GBS, but they didn't offer RAD-seq. So I'm still a little confused. It is definitely patented, and so you're probably going to be paying part of a license fee if you use it. But maybe if you just want to do it in your own lab, I'm not sure if you have to pay for it. So check into that. Don't come back to me when you're getting sued in 5 years by Fluorogenics. But this is how it works. So you have your genome, and you have these restriction enzymes that cut at these various sites. You select the fragments, and you actually ligate your adapters based on those restriction enzyme cut sites. And then from there, you sequence off of those kind of barcoded RAD ends, these restriction digested ends. And that allows you to consistently sample the same piece of the genome over and over and over. So you're building up coverage right along your restriction enzyme sites and not sequencing anything that isn't right by a restriction enzyme cut site. You can do single in or paired in sequencing. And when you map your reads back, it looks like this, right? So they're all lining up just perfectly along those cut sites. And so if you're looking for variation, you're building up coverage right here, and you're building up coverage right here. So if there's a SNP in between, you're not going to sample it, so you're missing data. But you're going to sample the same sites over and over and over again. So you're building up the coverage you need to adequately call SNPs. Versus if you had something like shotgun sequencing, where lots of sites might be covered by 1 or 2 reads, you're aggregating your sequencing into little columns, as little areas of high coverage, instead of trying to sequence everything. Does that make sense? Sort of. Okay. Yeah, so this would be an example where if you can have lots of coverage and you feel really confident you have a heterozygote, versus if you didn't have enough coverage, you might be a little worried, is that a heterozygote, or did I happen to get 2 random errors stacking up? So again, just trying to aggregate your coverage into the same area so that you can have the statistical power to call things accurately. The newest thing that we have used a little bit in trees is double digest RAD. If you actually have too many restriction enzyme cut sites across your genome, and you can pick your restriction and you can pick a number of different enzymes, but even if using a rare cutter, you're sampling too many sites and your coverage is still too low, you can use this DD-RAD technique, which was published Peterson et al. In 2012. And basically, you're going to use 2 enzymes simultaneously, and then use a very explicit size selection. And I have a picture of that. So this is like normal RAD sequencing, where everywhere that you have a restriction enzyme cut site, you're priming off of both sides, and you're kind of building up a little bit of coverage. Whereas here, the black X and the gray X are representing that you have 2 different enzymes, and you're specifically only looking for fragments that are between your 2 restriction enzymes and that are very much in the same size. So it's just another way to filter down to just a very, very small % of the genome that you're actually sampling. So in this case, only these areas that are right between the black and the gray, and that are in your size range that you're selecting for, are actually going to get sequenced. So you're able to just take what was even more too distributed up here and you weren't getting enough coverage and focus all of your sequencing efforts into these little tiny regions. And you can get down to where you're sequencing like 1/10 of 1% of the genome. But if your restriction enzyme cut sites are conserved across all your individuals, you're sequencing the same 1/10 of 1% across all your samples, which is the important part. I kind of liked this paper because it really, to me, set out a nice scheme for like what type of sequencing and what type of approach should you be taking based on your scientific question. So if you're in phylogeny or population structure, you may still be in the Sanger sequencing, or a lot of people are going to like exome capture or something where it's much more targeted and you want focused data in a few regions. If you're going to something that your recombination is limited, your LD is quite long, you don't want to spend too much money, you just want to sample a very small portion of the genome, but you need it to be random and distributed across the genome. So QTL mapping and pedigree mapping are both qualify for that. Because you have a population structure, your LD is generally much bigger, you need fewer markers. So DD-RAD is really good here. If you're doing something like association mapping or population genomic scans where you want a lot of density of markers and you need to invest more in the sequencing, you don't want to sequence the whole genome, but you want to get lots of markers, then you might be more in the RAD tag or the GBS. So kind of a fun way to think about tailoring your genomic approach to your question. What is the density of markers I need? How much money do I have? How many samples do I have? You have to ask all of those questions, and then you have to make really good decisions. Genotyping by sequencing is another kind of similar technology in this whole area. Elshire in 2011. They have a much reduced library protocol where it's much more streamlined, it's much more efficient, it costs less, reduced sample handling. They also were the first ones to really talk about using methylation sensitive restriction enzymes so that you're preferentially sampling the part of the genome that isn't methylated. So you're preferentially sampling the part of the genome that is likely to be expressed, holding genes. They have a slightly better barcoding system. And this is not licensed, it's free to use and sell. And this came out of Cornell. They used to have a really great service lab that would do this for you. We had a GRASS project here at UT, and we ended up paying, at the end of the day, I added up all the costs and divided it. It ended up being $46 a sample to get 96 samples done at Cornell. On their website for maize, this was their cost. We did not make it to $33, it was $46. But they claim that if you go to like 484 plex, you can get down to $9 a sample, which is really cheap to get 50 or 100,000 SNPs. So yeah, so it's great. And there are other places that do this as a service now. So the informatics for this is going to be a lot more challenging if you don't have a genome. If you have a reference genome, it actually can be quite similar to whole genome variant calling. If not, if this is something you're facing where you do not have a reference genome and you want to do this, the two pieces of software that work well are Unique and Stacks. I had a few students 2 years ago that were really interested in this, so we can try to work with those if you need to. Bisulfite sequencing, I wanted to talk about this a little bit. This is where you're interested in DNA methylation. Important to note, there's methylation on DNA. There's actually methylation on histones. This will not tell you anything about methylation on histones. This will only tell you about methylation actually on the DNA. But this is the first discovered epigenetic mark, and the methyl groups that are added to DNA are very interesting, especially when they're present in the promoter of a gene. They suppress transcription. So there's a lot of transcription regulation going on that even 10 years ago we knew nothing about. So if you're really interested in this and how it works, only cytosine is methylated in eukaryotes. That's all that we normally look at. But if you're in prokaryotes, I think adenine can also be methylated. So this is one of those clever illustrations of how scientists really took the short read sequencing and ran with it and said, you know, I can use this to answer my questions. And basically, if you bisulfite treat DNA, the unmethylated cytosines will turn into uracil. The methylated cytosines will stay the same. Do bisulfite treat, you convert all your uracils back to T's, I think. And then you sequence it. And then all of your C's that changed into T's, you know that was a methylated base. The problem with this is incomplete conversion. Bisulfite has a lot of DNA degradation, and while it's degrading the DNA, it does not always completely convert all of your cytosines. And so that can be a problem. But this is kind of the general idea, where you have a C that is unmethylated. Due to bisulfite, it will get changed to a U. With PCR, you're going to convert it back to a T, whereas this nice little methylated cytosine is just going to hang out and continue to be a C. So the take-home message here is just that this is great. This is an awesome app. None of the software we talk about in this class is going to help you with this. You need completely different informatics software. You need special mapping software. It has its own like downstream analysis for statistics and visualization. So depending on what type of data you're expecting to get for your research project, you may need to really dig into like what kind of informatics you need to do. Yeah, so I think I just said that. Many types of omics yield different types of information, and you need to tailor your data analysis protocols to your data production protocols. So very briefly, reference genomes. If you're not sure if a reference genome is available for your individual, there are a couple of places to look. First, the UCSC Genome Browser tends to only have the highest quality reference genomes, but they have really up-to-date annotation and usually a lot of annotation for each of your genomes. So 55 mammals, 27 vertebrates. I don't know why they only have one virus, but they have Ebola up there. So check that out. They don't have any plants. If you're in plants, don't bother to look because they aren't there. This is NIH funded, and it turns out NIH cares about everything on Earth except plants. Just very sad. Breaks my heart. But if you don't have a plant, you might want to look here and see if it's available. And this is a great place. Like they have just track after track after track after track for most of their genomes of all of these different layers of information. I liked this comic. 1995, we were like, yes, we have one genome. By 2000, we had 30. By 2005, we had 1,000. By 2015, we had 45,000. So really, lots and lots and lots of genomes being sequenced. If you go to NCBI will take all sorts of levels of genome, and people are usually sort of confused about what these terms meant, so I thought I'd go through them very briefly. Quote unquote complete genomes mean all chromosomes are gapless and have no runs of 10 or more ambiguous bases. I'm not sure I buy that chromosomes are gapless, but they are largely gapless. There are no unplaced or unlocalized scaffolds, so the people who submitted it believe that everything has been put in the correct chromosome location, and all of the chromosomes are present. So that's a very gold standard level. The next level down is the chromosome level assembly. There's sequence for one or more chromosomes, but there may be gaps. I think some of the most common genomes that we see every day, like chicken, are still in the chromosome category because they're huge 3, 5, 10 kb gaps where nobody knows what's in there. However, everything is localized to a chromosome location. From there, the third level is scaffold level. Some sequence contigs have been connected, maybe with paired ends or with PacBio or with mate pairs. So we kind of know how scaffolds are placed in relation to each other, but we don't know how they are located in relationship to actual chromosomes. So this is usually where you're seeing 1,000 to 20,000 pieces of the genome, and you don't really know what order they go in. And then contig is reserved for the least ordered and the smallest pieces. So genomes where we have tens of thousands to hundreds of thousands of little bits of DNA, and we have no idea what order they go in, and we don't even know how they're related to each other. So if you go to NCBI, pay attention. All the genomes will be placed into one of these 4 categories, kind of indicating their level of completeness. I went and updated this. It turns out that when genomes are shorter, they're much easier to assemble, not surprisingly. So if you go to the viruses, over 96% of the viruses' genomes are complete. If you go to the prokaryotes, about 7% are complete, and only 1.3% are chromosome level. And if you go to the eukaryotes, much fewer genomes, and less than 1% are complete, and about 13% are chromosome level. So there's a lot of resources out there. There's thousands of genomes, but many of them are not in super great shape. So today we're going to do examine some read quality and do some quality and adapter trimming. And the goals is to ask, is my data of sufficient quality to use? We've already learned that the instrument assigns a confidence value to each base. Are the bases high quality overall in my data? Does the complexity look normal? And this is going to depend on what type of sequencing you did, right? If you did 16S, it's not going to be very complex. It's all going to be 16S. If you did RNA, there are going to be genes that are very highly expressed, and you're going to see them more often than you see other genes. If you're doing DNA and you expect it to be completely random, you want to see very high complexity. But depending on that, you can detect if you had PCR or library prep problems based on the complexity. Are there adapters? Do I need to get rid of those? And you can also look at, are there lane batch effects? Am I seeing, on an Illumina HiSeq, there are 8 lanes. If you paid for sequencing all 3, you might see that lanes 1 through 3 were all failures, and 4, 5, 6, 7, 8 are all great. So you can start to look at, was there any instrument problem? This is what FastQC looks like. It's kind of a strange little program. It outputs things in HTML format, which is super weird. That's not a normal output format for bioinformatics, but it works really well. It is this 12-point quality analysis. You will never get all green check marks. Don't worry about that. We'll kind of go through each of these and talk about what they mean. But don't panic if you have lots of red X's and yellow exclamation points, especially if you're not doing a straightforward shotgun DNA sequencing project, because that's what it's expecting. But it still has a lot of useful information. Here's an example of the first plot you see, which is for each base, 1, 2, 3. It gives you the Fred quality scores. So we talked about those quality scores that indicate how confident the sequencing instrument was that the base call was correct. If they're all up here in the green, you know that these bases are very correct. If you start to see stuff like this, you know that lots of your sequences have probably been miscalled. So the difference between the two is pretty stark. This also shows you the typical problem with Illumina, which is everything starts out pretty high and then it degrades as the read gets longer, the signal reduces, and you get many, many, many more bases that have low quality values. Trimming. So after a quality assessment, you want to trim. From the quality control step, you generally know where the problems are. Do I have quality problems and do I have adapters? And your goal is to get rid of the bad data and keep the good data. And you may want to drop a read entirely if it's too low quality or too short. Today will be using Trim-O-Matic. It's a nice little piece of software. It's very flexible. It has a ton of different options. It handles paired-in data really well. It's threaded, so we talked about that a little bit a few weeks ago. That means it can use multiple cores or it can run 2 threads on the same core, so it runs faster. And one of the things when Trim-O-Matic came out, which admittedly was a long time ago, it was one of the first programs that actually did a really nice job of this. It detects read-through. So if your DNA fragment was really short, your side selection didn't quite work, and a few really short ones snuck through, and the sequencing read through into the adapter, and so you have the reverse complement of the expected adapter sequence, it will find that and toss it out for you. So that's nice. So all the different things Trim-O-Matic can do. It can cut your adapter sequences. It can perform a sliding window, which I think I have a slide on next. So basically looking at when that average quality degrades so much, it's time to just chop off the end of the read. If you know you want to get rid of the first 6 bases, you can do that. If you know you want to get rid of the last 6 bases, you can do that. Deleting and trailing are going to chop the front end based on quality. Crop and head crop are going to chop the front end not based on quality. And min length just says, basically, if you've done all this stuff and your read has dropped below 30 bases or 50 bases or whatever you want, it'll discard the read entirely. All of this is in the manual. So this is how, or the goal of having a sliding window is that you're sliding along. You start at the beginning of the read and you say, okay, good quality, good quality, good quality. I see one base that's low quality. I don't want to throw away my whole read. So I'm just going to keep going. I'm going to accept that I have one low quality base. And you keep going, you keep going. And then you see this one, you're like, I don't know. But then you start to see like the whole read is really starting to degrade. And so it makes this kind of intelligent decision to cut here. So keep the one bad base because you gain so much length by keeping it. But once you're really starting to get in this territory where you're not getting much gain, not many good bases are being retained in ratio to your bad bases, that's when it cuts. So it's a pretty smart program. Skewer is another program that we often use in my lab. It is faster than Trim-O-Matic, but a bit less flexible. So it doesn't have as many parameters to play with. Not necessarily a problem. And it's a very gentle quality trimming. So this is like the new thought in trimming. It's like we're throwing away too much data. We have to stop throwing away so much data. We need to keep more bases that are low quality. So this is kind of an evolving community wisdom that quality trimming does reduce error, but since it reduces your content so much, that can be a problem. And so this is kind of what I've come to the conclusion from reading. If you are mapping reads or counting, so doing RNA-seq, gentle trimming is fine because your reads are still going to map and you're still going to get your counts. And when you're counting, the most important thing is to retain as much of your counts as possible because like Daniel was asking, if you have really lowly expressed genes, you want to save all the data you can. Like that one read could be like the precious read that you needed to do your statistics. So gentle trimming. However, if you're doing assembly and variant calling where a bad base could cause really big problems, and we'll talk about why the miscalled bases cause really significant problems in assemblies, it's better to just go ahead and trim and do like a more stringent trim and get rid of a bunch of stuff. So depending on what your application is, you might pick one or the other. All right, that's it. You guys have any questions? That's a lot. All right. Okay. So we've gotten down to here. If you want to follow along on the website, we're no longer in software carpentry materials, so you have to put up with my little less detailed materials, but I tried to write everything down so you can still follow along and see what we're going to be doing. So we'll get to that in a minute. All right, so let's start by logging into the ACF, and I'm still logged in from earlier. So is everyone still logged in? We're still working on it? Okay. So we are going to go into your project folder for the class. And I always have to look up what that folder is. So CD, I think it's Luster, Haven, Courses, EPP, and then your personal user ID. Okay, so E. coli is what we're going to be creating. So I'm going to move that out of the way. I was practicing this over the weekend, trying to make sure everything was working. So you should have blast practice and script practice if you went through the materials that I posted for the day we didn't meet. Okay, so we are going to create a directory called E. coli and CD into it. C tab complete. I just type CD, E, tab. Done. Tab complete. It's your friend. Okay, so there is this whole system on the ACF called the module system that allows you to load software. Have I talked about the module system? Yes, I talked about it for BLAST. Awesome. So SRA Toolkit is installed, and this is going to be what we can use to download data from NCBI. Module load SRA Toolkit. And then we are all going to we're all going to download the same data. And people at the ACF are probably not going to be happy with me. We're going to do it anyway. Make dir raw data and CD into it. So I should draw this as we're going. We're gonna set up, I don't know if you guys remember, but I originally had a lecture about how to set up what I consider to be a quote unquote proper analysis directory. So we're naming our project E. coli. And we are going to have a raw data directory where you're going to put, not surprisingly, raw data. So the command is fastq-dump. Split files is a really important flag. If you don't split the files, it's going to take those forward reads and the reverse reads from your paired end sequencing and shove it all in one file. And the software we're using does not want them all in one file. It wants them in two separate files. So the split files flag says, take read one and put it in one file, and take read two and put it in a different file. And then we're going to give it the SRA accession number. And this will take about a minute. Now, we have not done the Q sub yet because the head node is the only one that can see the internet. So this part of the ACF is connected to the internet so that we can all see it and log in. However, none of these computers are connected to the internet, and that's a security thing. I don't know if you guys have noticed, they're very serious about security, the ACF. So if you're doing any downloads, you have to do it here, because once you move over here, you can't actually see the internet. Okay, so mine finished. I am going to copy and paste. I have put 3 wget statements. And if you go to NCBI, let me throw this over here. And I can't remember. So it turns out there are a lot of different E. coli genomes, I guess not surprisingly, because they've been working on E. coli. Researchers have been working on E. coli for a very long time. But this is the one that I've determined is, I think, the canonical, like, official genome assembly that people really care about. So if you go here, maybe here. Sometimes NCBI is just like, you just have to hunt around for stuff. Yes, here. So if you see this box at the top, this is really convenient. This is where I'm getting these links from. So it says download sequences in FASTA format for the genome. So if you copy that link and then come over here and do a wget. So if I do wget, and you can also just copy and paste it from the course website. Copy and paste it in. Oh. So copy link address. Yes, so that's how I found it. So if you're looking for your own personal genome, go to NCBI, go to the top. All of the stuff you want to download is linked in this little blue outlined box at the top. It's got your protein sequences, your gene sequences, your genome, your GFF. So for this class, we're just going to get these 3. This is the actual genomic scaffold. We are not going to learn about GFF files for a little while. We're going to go ahead and get it now. And then the protein sequences. So you can copy all of that and paste it here, and it'll do them all 3 one by one. Mm-hmm. The wget? Yeah, so that was from a previous lecture. This is the SRA toolkit. So NCBI provides a programmatic command line utility to suck data out of NCBI straight into your terminal. And it does that because transferring really, really big files over the web is very slow. So this command line utility is just really, it's just a very convenient way to get raw data out of NCBI. I think that was like maybe day 2 or 3 that I kind of covered that in the lecture. And it's really convenient. It only works for SRA data. It doesn't work for any of the other parts of NCBI. But for SRA data, it's really, really great. Okay, so I have got 2 FASTQ files. Hopefully you do too, an _1 and an _2. That's your first read and your second read. This is what we got FASTQ dump. I also have a genomic .fna.gz, genomic.gff, and a genomic protein.faa. So hopefully.
I don't know if anyone else is made mine is still waiting in the FASTQ dump.
Yeah.
Still hasn't come in.
Really?
Yeah. Okay.
Okay.
I saw a lot of other confused faces, so I just want to make sure.
Yeah. Okay. Let me. Okay, so everybody who's still waiting, Control-C. This is going to be, I think I want 755. I didn't teach you guys how to do permissions like this, but don't worry about it. The little plus minus system is way easier to learn. E. coli, raw data, *FASTQ. Okay, so everybody from inside your raw data directory, you are going to do, hold on. Copy Lester Haven courses, EPP, mStaten1, E. coli, raw data, *FASTQ to wherever you are. So that's gonna be, somebody try that and let me know if it works. Do you get a permission denied? Yes, use my username. Since it won't work for you guys, you can just copy it from me, right? Like I already downloaded it, so just grab it. Okay. Try it again. Oh, no. Man. Okay. Try it now. Does that work? It worked? Okay. Okay. Yeah, there should be 2. You should now see 2 FASTQ files. Did the wget at least work for most people? Okay, try that next. Actually, you can get them all right now. Copy everyone. It looks like you got yours and you have 3, 4, 1, 3, 4. You have 2 sets of reads. You got 2 different SRA sessions. Like the RRA 2, 3, 4, 1. So if you look that up in NCBI, did I get? I have 4 too. And those should match mine. These are something. Sure. I think that should still be copyable, but you guys won't be able to overwrite my files. I'm not sure. Oh, I was just saying Control-C if you were home waiting on. Yeah, so now I'm the LS. Most people have a bunch of files now, FastQs and GZs. All right, so next thing we're gonna do, that we might as well just get stuck doing, is this qsub command. And I'm going to copy and paste it because it's long. Fingers crossed. Still not working? Very good.
Yeah, the wget should still work.
Copy and paste wget.
Or if you just change that to star without fast key, get all of that. Which is fine too.
Yeah, I don't. Honestly, the internet has not been stellar in this building for the past few weeks. Everyone in my lab has been complaining about it. I don't know what's going on. Okay, I think I actually got, so the command uname is a command that tells you what computer am I on? Tell me the name of this computer. And if you just type uname, it says you're on a Linux computer. I'm like, huh, that's not actually what you really want to know. So uname-a, A stands for all. It's telling me everything about this computer, and it gives me a bunch of random information. But the most important one is that the name of the computer is not login. So I am now, we did the FastQ dump and the copying, and then I ran the qsub, and now I'm on one of these guys. So you guys are probably going to see other names of computers. So you might be down here or up here, like, or somewhere over here now. And let's just confirm. If I try to do a wget, it's just going to hang forever. So if you're trying to do a wget and it's not working, it's probably because you're on a compute node instead of login node. So to kill this, I'm going to do Control-C. You guys don't have to try that. I'm just illustrating the point that no internet. Did anyone else's launch? Are people getting their qsubs to launch? No. Okay, raise your hand if it worked. Okay. This is not bad. That's like half. I can live with that. I mean, it'd be nice if we could all get on, but it's better than last time. Okay, so for people who it has actually worked, you need to now, it has popped you back to your home directory. So now you have to CD all the way back to your project directory. You pull out raw data. While we wait for more people to get on, are there any questions? Great time to get your questions answered. Oh, I just like to illustrate that that's how you figure out if you don't know what computer you're on, if you're not sure if you're on a login node or a regular node, a compute node, the uname's just nice. And sometimes, so I have one terminal open and we've been using one terminal. You could have 4 terminals open and be on 4 different computers. And so for me, if I'm logged into like my server and the ACF and our hosting server, you know, I can do uname in any of them and figure out what is this that I'm doing here? Like, what is this computer that I'm using? So it's just a nice way to figure out more about like, who are you talking to? Where is this stuff going on? You don't have to. If the qsub launches, you know you're on a compute node. I just do it usually to confirm. That copy should work whether you're on a compute node or on the head node, right? Like the copy doesn't care because that's all the file system that can be seen from any computer. So if you queue subs and then you realize you're missing files, you can still do this copy. Put it up here. So this won't do anything for me because I'm just copying where I am. Where I am. If you did something like that, for me, it's gonna, you know, they're the same file. You can't copy a file to itself. But for you guys, if you do that, it's gonna copy all the files to your directory. All right, who's still waiting on queue subs? Okay, we're running out of time. Control C and just go back to the terminal, and you guys are gonna use the login node, and I will email the ACF and tell them we are about 50% this time, which is still an improvement. Okay, so one thing that we can do, oops, that's not what I meant to do. Okay, so I'm going to unzip all of those files that end in GZ. Let me do that by gunzip *gz. So now the ones that used to end in GZ don't, I'm not gonna worry about this one because I just downloaded it twice. So it doesn't really matter. I can actually remove it. Okay, so everyone should pretty much look like this. So it's a good idea when you download data to look at what you actually downloaded and what you're working with. One thing we can do is look at the number of lines. Might be quite long. So sequence data is generally quite big. So who remembers how we would determine the number of sequences in a FASTQ file if we know the number of lines? What did you say? Yes, divide by 4. That is right. And I think I remember how to do this on the command line. You guys don't have to memorize this, but I think this will work. Maybe not. Well, you can always do it on your computer. Okay, so 2,067,340 reads. This is a pittance by comparison to what you will normally get, but it'll be faster for us to use a kind of a small data set to get started. We can also do ls/lh and look at, so each of these files is only a gig. Again, small compared to what you could normally expect. And our genomic files are even smaller. So those are small enough and you could email them as an attachment and it would work fine. You can also do head. I always like to do this to make sure, does that look like a FASTQ file? Yes, it does. I see ats, I see pluses, I see sequences, and I see quality values. So that makes me happy. The other thing you always want to do when you download data, the tail is probably even more important than the head. And the reason the tail is important is because if something interrupts that download and you're only getting a part of a file, you can often tell because you won't have an entire sequence quatrain at the end of your file. You won't have 4, so you should always see the at, the sequence, the plus, and the quality. If you're missing 1 or 2 or 3 of these, you know that something happened and you're missing part of a sequence. So it should always end with that full 4-line sequence that's complete. The other thing, I'll scroll back up, and as we're saying, if you have a forward and a reverse read file, they should always have the same number of lines. If they don't have the same number of lines, again, probably something happened during your download of one and you need to try to download it again. We're not going to talk about GFF for a while, so don't worry about that one. But I will do a head on the genomic.fna and make sure, yeah, it says complete genome. That's reassuring. I can see that I do indeed have nucleotides. Looks like a FASTA file, so that's good. Okay, so now let's get to our quality examination. So in our type A personalities as scientists, we're going to do everything super organized. So I'm going to create a separate analysis folder and then name this one. Oh. So this is the structure we're starting to build. Or if we wanted to make this unwritable, so you could overwrite it and protect the raw data, you could do that. or if you're going to do your analysis over here, and when you inevitably screw up and start over, it will be very easy to just create another analysis folder and start again without messing with your raw data. Okay, so I'm currently in raw data. I'm going to back up and make that analysis directory. I'm going to cd into it, and I'm going to make a directory called 1fastqc. Again, I like to put numbers in front of things and do everything in order so that when I go back, I know exactly what order I ran everything in. And I'm going to change into that directory. Our module load system is, again, our friend. I can do module load fastqc. And then we can do, this is super easy to run. Okay, so let's go through this command. The command itself is just fastqc, so it's pretty easy to remember. I'm going to tell it to use 2 threads to make it go a little faster. You do not have to use this. You could just leave that out entirely. Next, it wants an output location. For the output location, I'm going to give it a single dot, which just means put the output right here, right in this folder I'm in right now. And then the final thing you have to tell it is what data do you actually, what FASTQ file do you actually want it to work on? Can you say that first? Yeah. fastqc is the name of the command. That's just calling the software. The first flag, the -t, is for threads. So the number is going to try to use 2 computational sessions or sets of instructions so that they can double up and run in parallel. So it's going to make it almost twice as fast. It's a pretty fast program, if you're, especially with files as small, but we're going to try it. You do not need it. The -o is for where you want your output to land. So you could put it in your home directory. You could try to put it in my directory. But no, what you want to do is put it in the directory you're in, so you can use that convenient little dot. Single dot means right here. And then the final thing is the actual, the location of the FASTQ file you want it to work on. The FASTQ file,
You've been, I mean, you've been saying the first time what file are you?
Yeah. Yeah. So every piece of software, you have to read the manual. You have to read the manual, and it'll tell you what order to put things in and where it wants all the pieces on the command line. And you don't get to change it around. You have to do whatever the manual tells you to do. There are some conventions, but they're not always followed. I mean, we've already seen some weird things today, like the -- split - files is, I guess that's a traditional flag, but I don't think it has a single letter flag to go with it. So like sometimes things are, you just have to read the manual. So this is, I think, gonna work. Let's find out if I did it. So while that's running, I will pull up the fastqc documentation. So hopefully you guys are seeing, it's telling you approximately how complete it is. If you Google fastqc, this is where you can go to a copy of the documentation. And every piece of software you ever run, you should go read the documentation. I actually can't figure out where the command line documentation is right now. I don't know. If you dig through all those links long enough, you will find it. This is really good because it tells you how to evaluate your results. So that is also worth reading. Yeah, so and this tells you each and every one of the things that we're going to see. It's somewhere with the documentation, and I don't know if it comes with a manual. So some things will come with a manual, some won't. So nope, it didn't come with a manual. So you just have to dig around on the internet. Did this work for everyone else? Yeah, okay. And now we can run the other one. Okay, so now while that's running, I want to talk briefly about secure copy. So it is very weird that FastQC creates an HTML output. I suppose it's kind of convenient because it's a nice way to force a visual display of a graph, but it's unusual. It's not something you'll see very often. We can't view an HTML output on the ACF. Well, you can, but that's a whole other, like, setting up that type of terminal and tunneling the web browser back and forth. It's a pain. We're not going to do it. But what you can do is copy the files back to your laptop, which it turns out you kind of want to be able to do that anyway, right? Like, somebody brings a hard drive, you have sequence data, you want to copy it up to the ACF, or you produce some results files and you want to copy it back down to your computer. So we're going to learn how to do that. So we've seen wget or curl, but that, of course, requires that the data be available on the internet. If it's just available on your local computer, wget's not really going to work. So the other two ways to do things are secure copy and FTP. You've probably all seen HTTP in web addresses. That stands for Hypertext Transfer Protocol. That is the protocol that runs most of the internet. It's what runs when you view an HTML page. That's how it's being transferred. It turns out there's a second and older, actually the original network protocol that's called FTP, and that stands for File Transfer Protocol. HTTP for web pages, HTTPS for secure web pages. So if you were on your bank's website, you should make sure you see the little lock symbol and the HTTPS. Good tip there. But if you're going to for an FTP site, that is for transferring raw text files. There are instructions if you are a FileZilla user. I don't know how many of you are. There's like this nice kind of GUI that you can launch on your computer where you can drag the files back and forth. And if you prefer that, that's totally okay. And there are FileZilla instructions for the ACF. But we're going to learn the fast, but a little bit harder on the eyes. It looks a little complicated, but it's really fast. So we're gonna learn the secure copy protocol. It's basically a command line interface based on SSH. So it's just like your terminal, only it has a slightly different syntax, but it's using the same underlying technology. So start by thinking back to the regular copy command when you're on a single computer. You're copying the location of what you want to the location where you want it. The order is always what you want. The next thing in line is where you want it. So copy from to. Secure copy is the exact same thing, except instead of the path just being the folder path, it includes your username, the name of the computer, and the folder path. So it tends to be a little bit more verbose. So let's say that I'm on my computer and I want to copy a file that's in my current directory up to the ACF. I would write scp file.txt. It's in my current directory, so I don't need to give it a path. Just file.txt. I want to copy it to mstain1 at datamover1.nix.utk.edu :tilde. That would copy it to my home directory on the ACF. And this breaks down as your username, the @ symbol, the name of the computer, a colon, and then the location to put the file. So it's still a simple copy command because you have your SCP and then the local file, and then the place where you're going to put it just has a lot more information than we normally have to put. If you want to copy from the ACF back down to your local computer, again, it's the same syntax, but reverse it, right? So secure copy, the thing we want is your username at datamover1.nix.edu.edu :/file.txt. So that whole thing with no spaces is specifying where that file lives. And then the very last thing, you can put a path, or if you put a dot, it'll copy it to where you are. So it's kind of like SSHing, only you're like rolling this all up into one giant copy command. Okay, so let me show you how this works. Or let's look at our results first, make sure we actually have some things here. So it gives us a .zip file, which has some files that we don't really care about. What I'm primarily interested in are these .html files. So I am going to open another terminal. So if I ls this is on my personal laptop. I'm not SSHing into the ACF, I'm on my personal laptop. And let's just make a directory called E. coli and cd into it. So if you want to be able to find this, actually it might make more sense. I'm going to go to my desktop so that I can figure out where this is when I actually want to double click on it. If you were on a Windows computer, it's like C:/user/username/desktop. I don't know. If you're on an Apple, you're just gonna CD desktop and it's gonna work great. If you're on a Windows machine, you're gonna have to hunt around a little more because I don't know exactly. Marilla probably knows. How do you get to the desktop? What's the path on a Windows? Oh, okay, so C? No. Oh, tilde/desktop. Okay, tilde/desktop, easy enough. I think that'll actually work on both. Everyone found their desktop? Okay. So let me pull this up. So from ACF to your local directory, this is the one we want to use. I'm going to make a directory called E. coli. Again, organization. Secure copy. Okay, just type which secure copy and make sure, did everybody get a path back? If anybody gets not found, red sticky note, and we'll get a secure copy installed on your machine. But it should have come with everybody's terminals. So secure copy mstaten1 at datamover1.nix.utk.edu : I have to put the whole location. The easiest way to do that is do pwd and copy this whole set of nonsense over here. Star HTML. So that is the entire, I wanna, from your username at datamover1.nix.utk.edu, that's the whole name of the computer, : no space, on either side of the colon, no spaces in this whole thing, and then the entire path to what you wanna copy. So in this case, it should look something like Lesser Haven Courses, EPP, your username, E. coli analysis, 1fastpc star HTML. And then I want to copy it right where I am right now. The bad news is you get to do a duo push again. And as soon as this finishes, I'll scroll back up so everybody can see it. Okay, so I hit accept and it immediately copied my files. And I see I have 100%. So the copy worked. So let me scroll back up. Here is that giant secure copy command for those of you still trying to type it in. Tab complete will not work if the file system is remote. It's so sad. I recommend copy paste. How many people did that work for? No. Oh, this is very sad. Hopefully it'll start working for people. How many people do not have an HTML file? Okay, no one raised their hand. So everyone's at the HTML stage, but no one knows how to move it to their own computer yet. So we're working on that. Yeah, so unfortunately, you can actually use the duo.acf.tennessee.edu. That will also work. You do not have to use Datamover, but they ask that you use Datamover. And honestly, if you go, there's like a whole bunch of these. They're called DTNs, data transfer nodes. So they actually live kind of parallel. This would be like Datamover 1. Datamover 2. So you can also log into these, but you can't get a compute node from these. You can only see the file system. So that's why we have slightly different, like duo.acf.tennessee.edu is like a login node, whereas Datamover is a DTN. These are details you probably don't care about and that don't really matter. They just prefer you use this server for transferring data. Anybody want to ask a question? Yeah. How many people are just having internet problems?
saying it's permission denied.
That's weird.
So I put this in, and it brings up. Yes. Very good. Is it 100%? That's not very.
Okay, so if you do LS/L.
About that error means that maybe the other one.
That is very few. What do you say about LS? Okay. And LS/L. Okay. Weird. You have read access. Oh, okay, Bonner. Login as your username. I think that's why, because it thinks you're someone else. Okay. Once you log into the ACF, so you're not actually logging in. It's just using your password to just transfer the files, and then it just disconnects you. It should at the bottom just say 100%, 100%. So let's see if we can get this whole thing to show. Oh. Yeah, so it should just give you the names of the files at 100%, and then if you else, like you're still on your computer. Like you're not logged into the ACF. It just that duo push was just to get permission to transfer the files. That makes sense? Okay. Yeah, you're not logging in. Log out. Okay, so go to your desktop. Like CD tilde / yes. Yeah, / do an LS.
Good job, Nicole.
Oh, how are you logging into the ACF? Okay, can you open it if you want to? No, that's okay. No, that's what this is though. You can access it from here. Do you have like PowerShell? Alright, stay for class and we'll work on getting you. I'll take that.
It's still showing.
Okay, so did all of this. Right. So what you would
That's right.
okay.
Exactly.
Use that one. But does that make sense, right? Like you're just in a different location. Yeah, so delete all the way back to Lester. And then change it like next. Oh. take the 2000 out. All right, so for those of you who it hasn't worked, feel free to stay. For those of you it has, just to quickly finish up the lesson, if you can get rid of all of the stuff on your desktop, you should now have an E. coli directory here. And it should have PASQ files in it, or it should have PASQC files in it. And you can click on one and it's going to open up in your web browser. And you should actually have quality statistics for your files that you downloaded. So, no, most people are not there. This is the goal, to try to get to here before the end of the class. If you can stay after, if you can't, just let me know. All right, so those of you who feel good, you've got it, go ahead and go. For those of you who don't, raise your hand. You're rolling out, try to like run around and help everybody.
It's the same like